the actual Earth, Environmental and Biological Sciences School as well. Faraj is a quiet and unassuming individual who has an unbelievable tenacity for science. And over the years that I've had the opportunity of interacting with him, I've just seen him grow in maturity and his scientific acumen just reaching such great heights. And I believe that he's got a bright future in science. He has an inquiring mind, and I see it at all our journal clubs. He picks up things that most individuals would probably just gloss over. His project has been quite a complex one. It involved looking at the structure, physiology, and molecular dissection of Tripogon loriformis. And the team that was around him, that actually guided him, was Tanya Shulaskin, Rizalcha, Brett Williams, and myself. And I can tell you that Akram Taji also played a significant role in this process because she was the individual that introduced originally to Tanya, and we got involved in it. Faraj had the opportunity of presenting parts of his work at 2 international conferences, the Gordon Research on Salt and Water Stress in 2012 in Hong Kong, and last year at the Interdrought 4 meeting that took place in Perth, and that is held in this region for the first time, and that meeting is normally held every 4 years. At both these conferences, he held himself in high stead. In fact, at the Gordon Conference, which is a very specialized conference on abiotic stress, specifically on salt and water, many of the colleagues came up to me to say, what a deep thinker, what excellent work, etc. So today you're going to get a glimpse of that. In 45 minutes, it's not easy to get everything together, but as Mark Twain said, it takes a long time to get a short presentation, to put something together. So Paraj, we look forward to you taking us through this journey, and all the best for you.
Thank you, Saradon, for your generous introduction. So thank you everyone for coming to my final seminar. The title of my thesis is Structural, Physiological, and Molecular Characterization of the Australian resurrection grass Tripogon loriformis during dehydration and rehydration. I begin with a background, structural physiological results, following by concluding remarks and acknowledgements. Everyone here knows how important water is for life. It has been involved in it, and it is dependent on it for all the metabolism and structural stability. 60 to 90% of all living organisms is made of water. And a small loss of water can lead to death. In our case, me and you, if we lose around 2% of our cells' water content, we could die. Plants are more tolerant. They can stand 10 to 45% water loss. Although the water is so important for life, it is not easy to find. It's scarce. Only 2.5% of the world water is fresh, and most of it is trapped in ice. Much of the remaining is also unreachable from our hand because it is deep in the soil. We cannot reach it, plants cannot reach it. However, due to because of the population growth and also the climate uncertainty that now we are facing, the demand for water is rising, and we need to grow plants with less water. Basically, we need to develop crops that can grow and produce results with less water. While most of the plants can be divided in 2 main groups of drought sensitive and drought tolerant, there is a small group of plants, land plants, that are considered desiccation tolerant plants. These plants can tolerate desiccation. They can lose all their water to air dry state and can wait until rain comes and they rehydrate and resume their metabolism, normal metabolism. So basically, the other 2 groups are considered desiccation sensitive and we have these desiccation-tolerant plants. Desiccation-tolerant plants themselves can be divided in two groups of fully desiccation-tolerant plants and modified desiccation-tolerant plants. Fully desiccation-tolerant plants are non-vascular plants, and modified desiccation-tolerant plants are vascular. Desiccation tolerance is common among non-vascular plants, such as many types of mosses. But among vascular plants, it's very rare. Around 400 species so far have been identified as desiccation tolerant. Resurrection plants themselves can be divided in two groups, whether or not they lose their chlorophyll, their photosynthetic apparatus during desiccation. So we have poikilochlorophyllus resurrection plants that lose their photosynthetic apparatus, and we have homeochlorophyllus that maintain their photosynthetic apparatus. One of such homeochlorophyllus resurrection plants is Trichogondoliformis, which is an Australian resurrection plant. It can be found throughout Australia, and on top we can see this plant growing in the field. And at the bottom, there are plants that we grow in greenhouse. On the left, we can see it in hydrated state, in the middle, desiccated state, and rehydrated on right side. This rehydration took only around 24 hours to take place, to resurrect. Because of that, many people call it 5-minute grass. Our aim is basically to observe this desiccation tolerance behavior and understand the mechanism that this plant uses to tolerate extreme dehydration, and hopefully we could use it to develop plants that could develop crops that be drought tolerant. This is our plant. It's a small plant. Each leaf is around 5 centimeters. And the reason that we choose this plant is that during desiccation, this plant responds with a variety of responses. Also, its availability, it can be found throughout Australia, and it grows in many types of habitats. It's easy to grow. It has a short life cycle, around 2 to 3 months it can produce seeds, although it is a perennial. It is a small, easy to handle in the lab. It's a true grass, which similar to many other major crops, major staple foods, such as rice, wheat, corn, and others. And it is diploid, unlike many other resurrection plants, and makes it easier for further molecular works. Now, if a plant, what challenges a plant faces during desiccation? They can be grouped in two major challenges: mechanical stresses and oxidative stresses. Mechanical damage is due because when the plant loses too much water, we have excessive cytoplasmic shrinkage. This shrinkage, because the cell wall does not fold in normal plants, can lead to loss of membrane integrity. The membrane can be torn apart. And the other problem is membrane fusion. When the membranes, different types of membranes get too close to each other, it can fuse, and it could be irreversible. Macromolecules need water, a film of water around them, or a coat of water in order to maintain their structural, natural structure. And if water is not there, these proteins could become denatured, which again is irreversible. For oxidative stress, when plants lose water, it can disrupt the electron chains in many types of metabolic pathways, such as photosynthetic pathway, and result in ROS production, reactive oxygen species. This reactive oxygen species itself can attack and disrupt other metabolisms inside the cell, and also it can damage other macromolecules such as proteins, lipids, and DNA. Resurrection plants have evolved different structural, physiological, and molecular responses that makes them able to tolerate desiccation. Now we look at some of these responses, starting with structural responses. The structural responses are to minimize stress, oxidative stress and dehydration rate, and to minimize mechanical stress. Reduction in leaf surface area by leaf folding and leaf rolling is a typical response which results in reduction of dehydration and reducing light damage. Cell wall folding, which is a unique response in resurrection plants and in normal plants in seeds, is to maintain membrane integrity. Vacuole fragmentation, which is followed with water replacement, also leads to maintaining the cell volume. For physiological responses, these responses are mainly to minimize dehydration and reducing photosynthesis. The stomatal closure is a typical response which, by limiting the gas exchange, reduces the dehydration as well as reducing the carbon dioxide that comes to the cell and basically choking the photosynthesis, reducing it. Pigmentation also reduces light chlorophyll interaction, and we have dismantling photosynthetic apparatus, which again reduces photosynthetic rate. Molecular responses, the molecules which are produced during desiccation, extreme dehydration, are mainly can be categorized in three roles. Whether they are regulatory, which themselves are signaling involved in signaling pathways, whether ABA dependent or independent, or they are transcription factors. Then we have detoxification role by antioxidants and proline. We have osmoregulation and structural protection through by proline, carbohydrates, and proteins. There are molecules that are shared in different parts, such as trehalose, which has also regulatory properties. Now, although there has been so much studies in the last century until now on resurrection plants, most of these studies have been limited in a small number of resurrection plants, mostly from Southern Africa. This study, this project aims to study a new resurrection plant from Australia, and hopefully this opens up a new window of opportunity to understand more about this unusual behavior. This project is divided in three parts: the structural, physiological, and molecular studies. For the structural part, we looked at general structures and structural changes. We looked at the general structures because this plant is largely unidentified, not characterized, sorry, it is uncharacterized. So we had to know first what is the general structure to understand the structural changes. For general structures, we looked at external structures from adaxial, abaxial, upper epidermis, lower epidermis, and internal structure, the anatomy of the leaf. For structure changes, I looked at the macroscopic changes, leaf folding, and microscopic, cell wall folding, and vacuole fragmentation. For this study, we used, I used a number of histological tools, experimental tools, which mostly these methods were chosen to have minimum impact on the structure of the plant during dehydration and rehydration, because I wanted to look at the structure of the leaf in their natural state. Most of the common histological techniques, such as most of the histological techniques have procedures that changes the structure, such as by staining or dehydration, rehydration, infiltration, in the solid medium. So I used mostly light microscopy, a freehand sectioning method that I optimized it for small and delicate tissues and both variable, and electron microscopy. The type of electron microscopy that I used was ESEM, or environmental scanning electron microscopy. In this type of electron microscopy, the tissue doesn't need to be fixed or coated. So the plant, the tissue in their natural state, hydrated or dehydrated, could be placed in the chamber and observed. Here we see some of the results from ESEM, which is the surface structure of the leaf on adaxial or upper epidermis and lower epidermis. On left, we can see that how the veins are located next to each other, and in lower epidermis also, and some of the major structures such as gland and prickle hair. In higher magnification, we can see there are some more of the structures such as papilla. We can see prickle hair, basal cell, somata, silica cells, and I'm not sure if you could see it, but the surface is covered with the wax, with epicutical wax, which is fuzzy stuff on the surface. On the adaxial surface, we don't have it. These structures are typical of plants from arid areas. Now to internal structure. These images have been taken from transverse section of the leaf using freehand sectioning. They haven't been treated at all, only not even staining, so these are natural color. The leaves are very small and tiny, only around 2 millimeter or less wide. So we can see that on top we have the hydrated state, and the bottom we have dehydrated. The leaf is made of bundles which are placed next to each other, and between them we have these buliform cells that during dehydration you see them dehydrated and the leaf falls. We have, in higher magnification, you can see some other major cells such as buliform cells, sclerenchyma tissue, bundle sheath, mesophyll, xylem, phloem. During dehydration, a thick pigmentation appears on lower epidermis. Now we look at the structural changes. So here we have a bundle from hydrated, dehydrated, and rehydrated state. The bundle has a typical Crohn's anatomy, and it has a mesotome sheet, which makes it XYME positive, which means xylem mesotome exists. I mean, there is mesotome. This type of a structure is typical of plants from arid areas again, and the biochemical pathway that it has is considered as NADME, which is the most water efficient type of photosynthesis among C4 plants. Looking at the dehydrated tissue leaf, we can see it has been shrunken, it's smaller. And we also can see the cell wall folding. I'm not sure if you could see it on the back or not. We have cell wall folding in the mesotome sheet, which hasn't been reported in other papers for this type of cell. Cell wall folding took place for other cells as well. We have vacuole fragmentation, which are these brownish orange color parts. Also, this hasn't been observed using in other papers because I mean they couldn't see the colors. They all used TEM. The other thing which is interesting is that during dehydration we have this scleroderma tissue on the abaxial surface, which when the leaf falls, it makes a shield around the leaf. Because scleroderma tissue at maturity is dead and a hard structure, it basically blocks the water, light to reach the chloroplast. On top of it, we have this thick pigmentation which blocks the light furthermore. During dehydration, we see again it's green, and our chlorophyll content, chlorophyll extraction showed that plant maintains some 70% of its chlorophyll content during desiccation. Now we look at the microscopic changes, mainly leaf folding. On top we have, well, these images have been taken by ESEM again, and on top we can see the leaf on upper part of the leaf from tip to middle of the leaf, which leaf folding is very tight. And on the bottom we see the base of the leaf, which leaf folding is loose. It's mostly like rolling, or is loose. Leaf folding results in reduction in leaf surface area, reducing light damage as well as reducing the dehydration rate. Also, it produces a microenvironment in the middle, which reduces water loss. I wanted to see what is the difference between this tight leaf folding and this loose leaf folding. So I looked at the internal structure and surface structure of the leaf from the tip to the base of the leaf, looking at this midvein. We have here tip, middle and base of the leaf, internal structure and surface structure. Here we have leaf folding which takes place is tight, but in the base it is loose. By looking at the structure, we notice that there is no scleroderma tissue present in adaxial surface of the midvein, where leaf folding is tight, and also on the surface we don't have hard structures such as prickle hair and silica cell on the midvein. But in the base of the leaf, we have this scleroderma tissue present, and there are other hard structures such as silica cells, as well as prickle hair and short cells. To summarize this part of the project, we can say that general structural features of Trichogonadoliformis is similar to the plants which are common in arid areas. The structural features and changes of Trichogonadoliformis benefits it by reducing the dehydration rate, which gives it more time to induce desiccation tolerance, and also it protects it against the structural damage and minimizes the light-induced damage. Sclerodermatous tissue and bulliform cells are involved in leaf folding, and scleroderma tissue has a significant role in protection of plant against light. For physiological work, our work has been, our observation was mainly on chlorophyll integrity, membrane integrity, and photosynthetic rate. Chlorophyll integrity, we looked at chlorophyll content and chlorophyll fluorescence using mini-PAM. For membrane integrity, we looked at electrolyte leakage using this conductivity meter, and staining by propidium iodide. Then for photosynthetic rate, we looked at the CO2 assimilation rate using glycol. So everything has been done in comparison to the hydration level. For hydration level, we'll first we start watering the plants until they reach the air dry state. It took around 6 days, and then we watered them and kept the soil wet for 3 days. During day 4 we have this large error bar, and the reason is that that's a transition day. We have 3 replicates, and each one of them is responding differently, because during this day they lost around 80% of their water during 24 hours. So because of that, the error bar is large. Then we looked at chlorophyll integrity, and in particular for chlorophyll fluorescence. Chlorophyll fluorescence is used as a benchmark to show the cell viability, cell integrity, because during the dehydration or stress, the ROS produced firstly damages the photosynthetic or photochemical pathways. And by measuring the chlorophyll fluorescence, it represents the health of the cell and the health of the chlorophyll. So basically we looked at 2 different parameters: maximum photochemical efficiency and quantum yield of the photosystem 2. The observation was pretty much consistent. I mean, the results were consistent for during dehydration and rehydration for both of them, except during the time that hydration was below 65%, which during dehydration, which leaf started to fold. When the leaf is folded, because we use minipam to observe it, the chlorophyll is not accessible because leaf folds and there are 2 barriers between light and chlorophyll. There is a sconcoma tissue, as I mentioned, it makes a shield around the leaf, and there is another thing which is pigmentation. These 2 stops the light to reach it. Basically, it's a technical barrier, which because of that we didn't have observation here. But apart from it, it shows that the chlorophyll fluorescence, the chlorophyll health was maintained, it was protected, or it recovered quickly during rehydration. From here, we looked at now to membrane integrity. Traditionally, electrolyte leakage is used as a benchmark to represent the membrane integrity, because membrane is impermeable toward electrolytes, and unless there is damage in the membrane, electrolytes are not released from the cell. So basically, when the membrane is damaged, it becomes porous, and electrolytes from inside the cell leak to outer space. And by measuring that, they showed how the membrane integrity is, in what level it is. Electrolyte leakage observation showed that at the beginning, again, before 60% relative water content, we had very low electrolyte leakage, and after this point, we had a very sharp rise on electrolyte leakage when it increased by dehydration. The way that during under 20% relative water content, some 2/3 of the electrolyte leakage increase took place. After rehydration, it's reduced. That is probably due to water that went to the cells and washed it out. It was surprising for us that electrolyte leakage showed that basically we have leakage from the membrane, and if we want to use it as a method that has been used previously in other observation, we should say that membrane is damaged. But our morphological observation showed that same leaves resurrected. So the membrane shouldn't have been damaged significantly. So we had to further examine the membrane integrity. So we used propidium iodide staining. Propidium iodide is a membrane impermeable stain. If the membrane is damaged, it can